An atom is about 1,000,000 times beyond the limit of what can be seen by a human eye. If you were to stack 6 magnifying glasses back to back, each with a magnification factor of 10, would you then be able to see the atom? The answer, frustratingly, is no. Even if you had perfectly manufactured lenses and stacked them in the perfect position, you'll eventually hit an impassable limit. So, what's the holdup? Well, visible light has a wavelength between 400 and 700 nanometers. If you try to magnify things below roughly the wavelength of the light you are using, the individual waves start interfering, and all you see is a blur. This is known as the diffraction limit, and is a firm roadblock prohibiting the imaging of anything smaller than about 200 nanometers with visible light. But you might have seen pictures of tiny cell parts, or even of atoms, which are on the scale of nanometers, far below the diffraction limit. This is possible by the use of electron microscopes, which leverage the fact that electrons have an effective wavelength 250,000 times smaller than visible light. So, we don't beat the diffraction limit, we just get a different threshold. In 2015, a group of researchers found a novel solution to this imaging problem. Instead of making better microscopes to lower the diffraction limit, just physically enlarge the details themselves to be above the diffraction limit. This is the foundational idea behind expansion microscopy, a technique where a polymer gel is introduced into cells, which then absorbs water and swells up greatly, increasing the size of all features of the cell. Then, a traditional microscope can be used for imaging, greatly reducing cost, increasing throughput, and allowing for a traditional contrast usage. One of the inventors of this technique, Fei Chen, was a graduate student at the time, and now runs his own lab at the Broad Institute, researching spatial and temporal genomics. I first met Fei playing basketball, and was thrilled when he agreed to talk about his work with expansion microscopy, his current research, and his research goals. Here is our full conversation. I really appreciate you coming out.
Yeah, sure. I actually am, I have a really strong, not a soft spot, but like a strong interest in actually converting folks from going from engineering or physics and into biology. Actually,
really,
those are some of the funnest people to work with. And I actually have a lot of undergrads, MIT undergrads or Harvard undergrads who's majoring in CS or engineering who come to my lab, and I try to advise them on how to do that and what my experience has been in doing that. So I actually, that's why I was interested in talking about
it. Thank you. Let's just get right into it. So you at CalTech undergrad, you were EE.
Yeah.
How did you, first of all, why did you choose EE back when you were, 16, 17, 18 or whatever it was? How did you get into that?
Right. I've always loved building things, tinkering with things and making them work. And that's for me, the part in WE that's the most fun. And I had a lot of fun doing electrical engineering at CalTech.
Were you hardware guy?
Yeah, I was a hardware guy. Yeah, exactly. And, all my roommates were at E's and we built a bunch of crazy stuff. And at CalTech, there's this tradition of this thing called Ditch Day where the seniors put on this basically this elaborate puzzle hunt for all of the underclassmen once a year, and in there's often very elaborate construction. We built this those Japanese video games where not video games, just Japanese game shows where you're playing it's human Tetris.
The shapes
you have to go into certain shapes.
Whatever.
We built one of those things, but it was with a projector, and then it sensed when people.
Oh, nice.
Anyway, so this be this elaborate day-long thing. So I really like building things. And that's E was really fun.
Yep.
But along the way, there was a strong community in electrical engineering at CalTech that was focused on building things with biology, synthetic biology. Those were early days of synthetic biology, and it's building circuits in cells or building circuits from DNA. And there was a lot of some folks doing information theory. How much computing could you do? Could you build a transistor with DNA?
Sure.
And I got really into that, and that's kind of what bridged me into biology. I think there's a group of people like Niles Pierce and Eric Winfrey and Richard Murray, and these folks were they're really engineers, but they were trying to build stuff out of biology, in particular out of DNA, and so that really like opened my eyes to doing more things in biology. So that's kind of how it happened, actually.
Okay. Yeah, as a fun kind of aside, there was a paper that came out a few years ago of using DNA basically as a particle physics detector.
Oh, yeah.
Yeah.
Was it from George's lab? Or it was like was it?
I'm not sure. I just saw it on archive somewhere.
I see.
I'll send you the link afterwards.
Yeah.
But it could basically just, whatever comes in and
changes, like mutates the bits.
Exactly.
Yeah, I saw George had like a paper, not a paper, or maybe like a bioarchive paper, like that for cosmic ray detection.
It might have been that one.
Yeah. No, it's a cool idea. I mean, DNA is like an amazing substrate, right? Because we have these like really honed nanomachines that evolved over billions of years to let us manipulate them. It's like that's why people are excited about. I mean, it also had this crazy rich, dense information density, right?
Yeah.
And DNA storage and DNA computing. I mean, there's some pretty exciting things.
Yeah, for sure.
Yeah.
So, okay, you were doubly, and then eventually you ended up at MIT in Ed Boyden's lab. What was there a learning curve for you on the biology side, or was that pretty easy for you?
I mean, this is why, as I said, I'm really interested in getting folks who are passionate, excited about biology from the outside, maybe in, because there's so many different paths to get there, right?
Sure.
And you could be coming from physics or engineering or CS, and people often ask like, oh, what like textbooks do I need? Like, what should I like? What classes do I take to learn about biology? And you asked about this learning curve, and I think I was lucky to have realized that in the middle of my undergrad that I wanted to do this, and so I took a decent number of courses, but looking back, I mean, when you think about it. A lot of courses aren't, a lot of courses about teaching you facts.
Yeah.
And certainly that's not how engineering or physics courses are, right? They're like teaching you how to like derive something from first principles.
Yeah,
right?
exactly.
And I really like looking at the world from that way, but it's hard to derive biology from first principles. And, but, there is a level to that, right? Like, I think it's not that important to know the facts, but what you need to know are like, what are like some concepts that are really important, right? Like the central dogma, or like these sorts of things in biology that often come up, and you can imagine making a course. I mean, I'd be interested in making a course. It's like a crash course in biology, right?
That'd be pretty cool. Yeah.
For engineers and physicists.
Yeah.
And there's a lot of ways to like translate how you think about biological concepts into the language of engineers and physicists too, which I think will be very interesting. Because often those courses, if you take a intro bio course at MIT, I haven't taken it, but at least at CalTech, it certainly isn't taught in the same language, and that's a really technical school.
Right, absolutely.
And yeah, so the transition. So there's two aspects about the transition. There's like the knowledge, and this is true for anything, right? There's like the knowledge and the practice, right? The practice, the knowledge is easy to pick up, but the practice is like intuition about how you do experiments and how you think about questions. Mm-hmm. That takes a long time, I think.
Yeah.
And it'd also be great to think about how you can impart that to people in a more, shortened timeline.
Yeah, right, for sure. So kind of along those lines, when you well, first of all, how big is your group? So you have the Chen Lab over the road, right?
Yeah, my group is about 15 folks.
Okay.
And it's about 5 graduate students, 5 postdocs, and 4 or 5 staff.
Okay.
And yeah, I mean, the graduate students, they all have like pretty diverse backgrounds. 2 of them have a background in biology, and then the other 2 some of them have backgrounds in engineering, right?
Sure.
Like pure CS or math.
Yeah.
And I think graduate school is a good time to make the switch. Some people also try to make the switch during postdocs. I have a postdoc in my lab, and her background was in like astronomy and in for PhD, and then like in biophysics for astronomy in undergrad and biophysics and for PhD, and so and there's a lot of skills and that translate over. We do a lot of microscopy, so
aware. Yeah. How important one thing that I've seen in undergrad is a huge emphasis on kind of theory and tests. And then when you get to grad school, at least if you're at least casually involved on the experiment side, knowing how to actually work in a lab and like knowing the actual experiment is a completely different set of skills.
Right. Exactly.
What do you look for when you're looking for an undergrad? Like what kind of stands out to you? Like, what are you looking for in kind of a set of skills?
Yeah. I would say that you're right. There is this focus on theory in undergrad, and I do really think it's important to be exposed to lab environments, and I think MIT does that really well. CalTech does that really well, where there's these built-in undergraduate research programs. I think there's just an aspect of thinking about problems, like thinking about how to plan and like debug things when things are going wrong that you can't pick up in a theory class. Obviously you need the theory. And for undergrads who come to my lab, I'm actually just looking for folks who have interesting backgrounds, like, and who are pretty passionate, like who seem like they're like excited about.
Yeah, sure.
And I've taken some undergrads who've never had it, like at least a couple of undergrads that have never had any experience in biology.
Sure.
Like a pure CS guy, and who has an interest in them migrating over to the wet lab, and we talk about questions like, how do you think about the problem? How do you debug when things are going wrong? And I think that's actually really valuable for them. And even though they don't, I think these folks who are transitioning, it's super valuable for them. Yeah. Even if they don't really accomplish like any of the main goals of that project.
Yeah.
And I think it's valuable for the people in the lab who are mentoring them too, because they get to see that, like, how to teach someone about the concepts of the project who's like, looking at it from a completely different perspective. And sometimes you get insights, right? Like, why don't we do it like this? And you're like, well, I don't know. People have always done it, this way. And it's an interesting. I think it's good for both parties, even though, not much like. Like people think about projects as milestones. I think, if you don't think about that, like there's like no like real milestones have been met in those projects. But I think a lot of like, thought and just changes in way of thinking happened, which is great.
That's really interesting. So it sounds like now you're kind of more of like a mentoring role than
for sure.
How do you feel about that? Because it was only, a few years ago or so that you were a grad student, right?
Yeah, exactly. Yeah. I mean, mentorship is great. I mean, as I said, I really enjoy this aspect of mentoring people through the process of figuring out biology. And obviously I also have lots of folks who have a background in biology. And I think the best part about mentorship is, you get to see people really like come into their project, right? Like they probably start very green, and then you can see the growth, and it's often very rapid, and I think that's pretty gratifying. In terms of like learning how to be a mentor, I mean, it's like hard job. I think you have to pick it up on the fly. Yeah. And I think that the hardest part about that job is like every person is pretty different. Like there's not like there's no algorithm. You can't like just like plug people into the algorithm. And I think you maybe learn like archetypes. Maybe there are like groups and different types of people, and you get to learn how to optimize your mentorship for that type of person. But I'm still learning how to do that, I think.
Yeah. No, I'm not sure if you ever finish learning it.
It's a hard, but I think it's a rewarding experience.
Would you say that's kind of the hardest part of your job now, is figuring out the mentorship stuff, or is it kind of technical aspects?
No, for sure. Yeah, I mean, I think that is definitely that's definitely the part of my job that I spend the most time on.
Okay, interesting.
And it's hard in the sense that it takes a long, there's a long arc to it, and you kind of have to figure it out as you go along.
Okay, awesome. I want to get into your current research, but before we do, I hope that it's okay if I bug you a bit about expansion microscopy.
Yeah, for sure.
So, I learned about this just like, I don't know, a couple months ago or so.
Yeah.
It's the sort of thing that I was like, oh wow, like how did no one else think of that? Could you, give like a quick description of what it is and
sure.
How it works?
So, expansion microscopy solves this problem in microscopy of how do you take images beyond the diffraction limit, right? Because of the wave nature of light, you can't resolve features or that are closer than maybe 250 nanometers apart, and
that's just because like the wavelength of normal light
is maybe like 500 nanometer.
So then the waves start kind of like overlapping.
Yes, exactly. Okay. And so you the limit is like the wavelength over 2, basically.
Okay.
So and the 250 nanometers sounds pretty small, but if you think about biology, like a lot of really cool stuff happens at 250 nanometers. Like a mitochondria is about like 1 micron wide, and then a synapse is about like 100 to 200 nanometers. So if you wanted to look at, how the proteins are interacting within a synapse, you need to go and look at it at greater than the diffraction limit. And so this has been tackled with like clever and really cool engineering on the optics side or on chemistry side by other folks, and those people. There was a Nobel Prize that was awarded just a couple years ago on that. But those sorts of approaches generally didn't scale to tissues where there was a lot of autofluorescence. Sometimes you had to do single molecule imaging in those cases, and it was technically challenging. And so we kind of had this idea of, it was a crazy idea at the time. You say that right now, I think right now we say, oh, how has no one like thought about this before? I think before it happened, the idea, when you propose the idea to people were like, oh, that's not going to work, right? So I think there's this kind of inevitability to it when you when you see it from the other side.
Yeah.
But before you get it working, there's kind of like there's a lot of this doubt about like, oh, is this ever gonna work, right? And,
yeah, sure.
And the idea was just like, instead of making better microscopes or like coming up ways of like beating the diffraction limit that way, what if we just made the features, like if we separated out the proteins in the synapse so that they were further away from each other, then you would be able to resolve them on a normal microscope. Now, if the point is to measure like the spatial organization of things, which oftentimes is the point of microscopy, although it doesn't have to be always, you kind of want whatever this process that's like separating them to be isotropic spatially, right? Or like to preserve the relative positioning.
Sure.
And that was actually the challenge.
Yeah.
So how extension microscopy works is that you take some cell, let's say you have a cell, and these are dead cells that are fixed. And what this fixation process is a chemical cross-linking process that basically preserves the location of proteins. It comes from like embalming. Actually, people used to embalm. Not like, you know, if you go to like this is a little bit more, if you go to a morgue, people are like embalmed in like paraffin formaldehyde. That's like really strong smell.
Yep.
And that's the same stuff they use in this fixative.
Huh.
And it chemically cross-links the proteins such that the cell is dead now, but it's still retained in the position of things are retained.
Kind of like frozen sort of thing.
Exactly. Yep. And so you take the cell that's been like processed like that, and then you, what you do is you form a hydrogel inside of the cell. And how this hydrogel works is that you diffuse in these monomers, and it's in situ polymerized into this network of polymers. And the key special thing about this hydrogel is that there is this monomer group that's quite charged, and when you form it in high salt, the charges are shielded, but then when you wash it out in low salt, you get this osmotic swelling because there's these charges that are fixed in this network.
Yeah, sure.
And you get this like strong osmotic pressure to balance out the relative concentration of those charges. And what that manifests as is this physical expansion of, you know, the polymers of the hydrogel, when they're formed in high salt, they, just like entropically are like in this tangled, configuration. And then when you put them in like low salt, there's this osmotic swelling, you stretch out the polymers. And physically that manifests as something, if you cast one of these hydrogels, you can get like a 100x increase in volume. Wow. And this stuff has been actually really well studied here, for a long time. And actually here at MIT Chemistry, since the 80s, these, super absorbent polymer hydrogels, and they're used in different sorts of applications. The famous one that people always talk about is like baby diapers, but
sure.
They're used in a bunch of like real world materials applications. So we co-opted this hydrogel to physically swell the tissue or cell. But the main difficulty is that you can't, if you just think about a piece of tissue and you wanted to like stretch it 5x in each direction, you might expect there's some resistance to that, and it might tear or break or these sorts of things.
Sure.
And in fact, when we first did expansion microscopy on cells, you saw that. You saw cells getting torn apart. And the rest of the process was actually figuring out how to basically cross-link the proteins onto this hydrogel network, connect them to the hydrogel, and then deconnect them from each other so that you're forming this cast.
Huh, okay.
And then you expand the cast. And then that turns out to be very isotropic. It's the isotropicness of that is governed by the mesh size of the polymer network.
What do you mean by that exactly?
Oh, so this polymer is formed through this polymerization process that kind of connects up these monomers with cross-linkers, and you get some random statistically governed distance between how many monomers are between each cross-link.
Sure.
And you can imagine each a group of 2 polymers connected up to 4 cross-links, or 4 polymers go up to 4 cross-links forms a square, right?
Sure, yeah.
And the mesh size is maybe something like the characteristic size in space of that square when you're forming it. And people have characterized this for polymers of this type, and it's something like 5 to 10 nanometers.
Okay.
And you would expect that if you got really close to this mesh size, this grid size, you would incur errors in the expansion process because, they're like, you'll just get a lot of like statistical fluctuation between how many monomers are between each point, right?
Sure.
But that gets averaged out at length scales much larger than that.
So basically, because your mesh size is on the order of 5 nanometers and you're hoping to resolve things that are on the order of like 200 nanometers,
right. So
it's not a problem.
Exactly. Actually, we try to resolve things. So what happens is that the tissue gets physically expanded, and then now what was 200, like what was 50 nanometers is now 200 nanometers.
Right.
And so now we'll see things at the 50 nanometer length scale.
Yeah. But just still a lot smaller than 5 nanometers.
Yeah,
so
exactly.
The isotropicity, I don't know. The fact that it's isotropic still holds in that regime.
Yeah, exactly.
Okay. One other question about this is, is it important at all what exactly the expansion factor is? Or not really?
I mean, it is important depending on what you want to do, in the sense that like the expansion factor kind of sets your resolution. And then there's some physical constraints to the like hydrogel towards expanding.
Okay.
The maximum you could probably expand it with one hydrogel is about 10-fold in linear dimension, which is 1,000 times in volume. Right. So, I mean, if you just think about that, like the hydrogel is 10% or like something on the order of 10% weight, weight when you form it.
Right.
And 1,000 times in volume would be like, now you're at like 0.01 or 0.1% by weight, right? So like most of this thing is water now.
Yeah, wow.
And so that's like the physical property of the thing sets that factor.
Right.
We settled for something like 5x expansion.
Okay.
Because of both the mechanical properties of the hydrogel and also kind of the resolution we wanted to hit.
Right.
Subsequently, I mean, there's been a lot of work that's developed versions which are like about 10x expansion, or there's these iterative, like you can imagine expanding it and then like casting another hydrogel into the and expanding it more to like 20x and that's something that the reason why a lot of folks are into that really high resolution is that there's some ideas about using, and we were very interested in, I think Ed's group is still very interested in using it for connectomics, where you might want to approach like EM level resolution, and which is, you want to approach the sub 10 nanometer resolution, right? And there you might need 20x expansion is 10 nanometers, right?
So this might be a silly question, but would it be useful at all, once you do the expansion, to then pass it through like an electron microscope, or would you not gain anything at that point?
Yeah, I mean, it is interesting. It could be useful. I mean, the thing about the electron microscopes is that they work. There's a couple of modes they work in, right?
I actually don't know much about electron microscopes.
I
know the Compton wavelength of the electron is like a 1/10 of a nanometer or something. Yeah, exactly.
So your resolution is huge.
But that's about all. I don't know the actual, working with them.
Yeah, well, there's like, you can get, and I don't know that much about electron microscopy either, but basically you can either get like absorption, or like basically like things that where like electrons don't pass through, and that's like metals, or that's like how you generate contrast, right? It's like hard to generate contrast to electrons. Like normal biological structures don't generate a ton of contrast, right. And what people are doing is that they're like staining the samples with stuff like osmium, and that's like this metal, and they found out this formulation that like stains the metal into like the membranes, like the lipid bilayers of cells, and that's how you generate a lot of these EM images for connectomics. I think there's some idea about doing that. I mean, obviously, electron microscopy images are already way past the diffraction limit, so that's why it might be not as interesting to do it.
Yeah.
Then you might ask, why don't people just use EM all the time, right? We already have this instrument that, and the reason is that actually it's this exact thing about contrast. In fluorescence microscopy, we can generate contrast on like on specific proteins, right? We can fuse a protein to a fluorescent protein,
Sure.
or we can generate an antibody with a fluorophore. We can connect the contrast that we're generating in the image to specific molecular features, which is really powerful if you want to like label proteins in the synapse, or if you want to look at like DNA or these sorts of RNA, these sorts of things. But in EM, you have these like brute force tools for contrast where you're staining with some heavy metal or some other thing. And so you stain lots and lots of things. But I think there is kind of a blossoming field in making new EM contrast agents, right? Like what if you could make contrast agents that labeled a very specific biological feature, right?
Right.
Yeah.
Oh, I didn't know anything about that. That's pretty interesting. I wanted to ask you, because I'm, obviously a grad student now, and was reading about how, you were a grad student when you were kind of working on this. What was it like kind of on like the, human side of it or personal side of it, of being kind of on an exciting new discovery back in grad school?
Yeah, it was very exciting. I think I remember I worked on this with another graduate student, Paul Tilburg. And we, we didn't really know if it would work. It seemed like kind of out there.
Yeah.
But we did this first experiment, and that's actually a good thing to maybe learn about biology, is that in the first experiment we we were imaging this thing at like 3 a.m. and like we were like, oh wow. We actually were imaging it as it progressively got bigger, and we were like, oh wow, this thing is this cell is getting a lot bigger, right? This thing might work. And I think one lesson about that is that you might want to optimize for like doing experiments where you see large effect sizes in the beginning, right? Because then you might know it's robust. But yeah, it was super exciting. I think being a graduate student is sometimes a lot simpler too, right? You have ideas and you are excited about them, and then your whole job is to go and implement them. You spend your time thinking about it, and you didn't think that much about, like, we just we had this idea and we tried it and it worked, and we wanted to share it with the world. It was like a very simple process. I think a lot of times science is more complicated than that.
Yeah, sure.
And looking back, that's like one thing that I really appreciate about that process, actually.
What do you mean about the process of your thesis work was kind of straightforward and worked out, or just like the general process of science being kind of difficult?
Oh, I think it's just that there's a lot of aspects of science that are not as fun as doing that, right? Like there's writing grants, or there's like as a PI you often talk about like the things you want to do,
right?
To other people, and you try to convince them, oh, this is going to be a cool thing. But if you look back on that process, it was like, oh, this is something I want to do, and I did it, and then it was a cool thing, right? So so that's what I mean in this sense.
Okay, got it. Yeah, no, it's pretty. It was pretty cool to read about, honestly. Was there? It sounds like maybe not, but was there any particular kind of sticking points on making the whole thing work?
Oh yeah, definitely. I think there was. When we first did the experiment, the cells were getting bigger, but then you see these like artifacts, like if you stain the microtubules, which are like the kind of, you can imagine them as like the skeleton of the cell.
Okay. Yeah.
And they provide like the mechanical rigidity to a cell. You can see that along the orientation. This is actually pretty interesting. Like along the orientation of the microtubule, like imagine if you had like a bunch of like strings, right? When it was expanding, you actually saw these like stress fractures, right?
Oh, wow.
Where like the fractures were lined up across all of the
That's pretty
the
cool.
microtubules. And obviously that's like a really cool, like you might be able to use that to study like something about the force.
Yeah,
right?
that's what I was thinking.
Exactly. But, you know, at that time we were like, oh, well, this is obviously not good if you want to make turn this into a microscope.
Sure.
And so there was a little bit of engineering and thought to figure out how you got around that part. And that's this idea of like making this cast where you transfer the molecules into the hydrogel, and then you use like what we use is like a proteinase. It just like cuts out proteins so they're no longer cross-linked to each other, and that basically removes the mechanical force of internally to the sample.
Yeah, got it.
Yeah.
Okay, cool. Yeah, so that was your graduate work basically. And then could you walk me through what you did, from grad school to now?
Right. Yeah. So I graduated, and then I went to the Broad as an independent fellow, and that's kind of this thing that's been around and it's getting more popular in biology, which is oftentimes like biological training is long. You do a PhD and then you do a postdoc for another, the same length as like a PhD.
Right.
And there was some idea basically of trying to get people earlier in their careers to go into being able to do their own independent research, which is I think a noble idea. And so this was maybe even pioneered here at the Whitehead with the Whitehead research fellows.
Oh, wow.
Like in the 80s or something. It's becoming more popular in recent years, and so the Broad had started its first fellows program, and I went as a fellow, and all that means is that you get some funding and some space to start a small lab, and so I did that, and the problem that I was really interested in when I started my lab is that I had been developing these microscopy technologies for a while in Ed's lab, and it was clear that microscopy is really powerful, but like there was this limit in how many. So we talked about the diffraction limit, which is like the limit in how many the resolution. Mm-hmm. But then there's also kind of this there's this limit in how many things you can look at on the microscope at a time, because basically we use fluorescence, mm-hmm. and if you just think about how many colors you can separate out from like the optical spectra.
Yeah.
That's like the limit in the number of things you can label.
Right.
And
so like 6 or something like that.
6 or something. Yeah, exactly. And so, but there's like 20,000 genes, right, in the genome, I mean, maybe more. I mean, there's lots of like variants of genes. Anyways, there's at least 20,000 things you want to look at.
Yeah.
And so, so that was the challenge. I mean, and on the other side of this equation, what the Broad has been really good at, before, in the last decade is taking samples up and like taking samples and grinding them up and like looking at the relative abundances, like the compositions of these 20,000 things, right? Okay. Without any spatial information, but you can go and that's what the process of sequencing kind of is so you can go and like, at least for RNA sequencing, you can go look at the relative abundances of these 20,000 RNAs. And so like my lab was really focused on marrying these two worlds, right? Yeah. Like how do we look at, the diverse array of biomolecules that you can get by sequencing, like, putting it onto a next generation sequencer, but doing that inside of tissues. And so, so we did a lot of technology development in that front.
Rolling it back a little bit, when you said like you want to measure the relative abundance of various parts of RNA or parts of genes, could you tell me, so what I think of back to like my chemie days is if I want to know the relative abundance of something in a sample, I would go to like a gas spectrometer or something, mm-hmm. and I could see like elements. So could you tell me, just because I'm not a super bio guy, so I don't know, how is that kind of traditionally done of measuring relative abundances?
Yeah, that's a great thing. So you can spectrometry is still used, right? Like if you in particular, like mass spectrometry is used all the time to measure the relative abundances of proteins.
Okay.
But what has really taken off, I would say, in the last 20 years is measuring nucleic acids like DNA and RNA.
Okay. And
it's kind of interesting. It's because you have enzymes which let you copy DNA and RNA. That's really what's like at the crux of it all.
Okay.
Because if you have a protein, you have like one copy of that protein, and you have to be able to go and make all your measurements on that one copy of that protein. Right. But if you have a piece of RNA or a piece of DNA, you can copy it, right? Yeah. And you can take one copy and make it into like, a million copies. Yeah. And then you can easily measure that million copies. Right. And so that was the first step. So what happens then is that you basically can use the sequence information, right? Like each gene can be represented as a unique string of A's, C's, G's, and T's. Right. And so you just need to go and count the number of strings of A's, C's, G's, and T's if you want to figure out the relative abundance of a given gene. Right. So maybe taking a step back, in each cell there's one genome, right? Yep. And that genome has like 3 billion letters and is, and has like 2 copies on average of every single gene. Yeah. And you really need to know the relative abundances. There, what we first did was we went and sequenced or like determined the relative order of A's, C's, and G's and T's in the genomes, right? That was like the Human Genome Project. Yeah. And we did that in like around 2000. And it cost about $1 billion to, that's actually not that bad, right? Like, I don't know how many real dollars like today would be, right? Yeah.
Well, it's a lot less expensive than our particle physics.
Yeah, or like James Webb Space Telescope.
Yeah.
Probably, yeah. I mean, that telescope's amazing, but like, it's not
billions.
That. Yeah, but that's one human genome, right? There's a lot of humans out there, and one of the next things that people did was like, how do we look at the variation, right? Like the variation in my genome versus your genome, right? And how do we get a sense of that variation across populations? Yep. And what we did, there was an intermediate technology that just looked at the point, like it was a comparator basically. You could just compare, if we know like there's about 0.1% of the genome is like maybe different, and like some fraction of that is relatively important, we can just like take all of those points and compare you and me. And that was like a less difficult problem than going and sequencing all of the genome between your genome and my genome, because we already know that like 99.9% is the same. So why would we like go and do this again? Yeah. And so that's how we collected a lot of this information about like human genetic variation. But then there was this kind of revolution in sequencing technology, or the ability to reconstruct A's, C's, and G's and T's from samples. That was really led by this company that's now like, dominant in the field called Illumina. And basically what they did was they made a very high throughput fluorescence microscope, and they gave each base like a different color. Okay. And they engineered this enzyme that and a set of bases that would like add one base. It's like a combination of enzymes in chemistry where you would add one base and then you can image it. Okay. And then you can add another chemical to like cleave the dye and then add the next base. Yep. And so you can just take these videos. Wow. And then you would lay out like, millions of these molecules on like a flow cell,
Yep.
and then the microscope just goes in and takes millions of these videos. And so that like basic idea drove the price of sequencing from, $1 billion for the human genome to maybe it costs $500 to sequence a human genome right now.
Right.
In like 15 years.
Yeah.
And so there was this like super exponential decrease in the cost of sequencing.
Mm-hmm.
And then that made people realize that it was then possible to take samples. And so every cell has one genome, and that genome is converted into messages to make proteins. They're called RNA, like mRNAs, messenger RNAs. And so like those are so if you think about, you can think of those as the instructions. Like that's like the analogy. People like the instructions for the cell, right? Uh-huh. And so if you think of the cell as like a state machine, that's like another like way, like people like, that's another like engineering now. You think of the cell as like a state machine, you can like probe the state of the state machine by looking at the instructions that's currently in it. And so people realized that one way to do that was to go and count the different like instructions, or like figure out the relative compositions of the instructions.
Yeah.
And like what instructions is the cell executing, and like in what proportions are the instructions being executed.
Right. Okay.
And so that's basically RNA sequencing.
Mm-hmm.
And it turns out that you can use the same chemistry that's revolutionized DNA sequencing, but now you sequence RNA. And now instead of trying to just determine the sequence, you can actually count the relative proportions, right? I saw this RNA like, 100 times, and I saw this RNA one time.
Yeah.
Right. And so that's really been able for us to like look and dissect out like states of cells, right? It gives us a way to connect. It gives us a way to connect basically like genes to like the functions of cells, or like what cells are doing.
So could you, like, for example, like turn off a gene and then you'd be able to notice that there's a decrease in some mRNA?
Yeah, definitely.
Okay. Oh,
that's
interesting.
So like another way to, one thing that I think is important, like if you think about like what it would be important to go if you want to learn about biology, coming from an engineering background, is that you might want to know about like characteristic numbers, right?
Yeah.
And
right.
like, you want to know the order of things.
Exactly.
And so it's important to remember there's 20,000 genes. It's important to remember maybe that there's this website, BioNumbers. That's a great, it's like a compilation, people publish a bunch of papers, and they measure different constants, these for numbers, and it's a compilation of these like characteristic numbers. And so one thing that you might want to then remember is that like an mRNA has a half-life of like a couple of hours in a cell. And so it is in fact like a transient state you're measuring. So and maybe here's a good way to illustrate that analogy, right? Like there's two things you can think about. You can think about different, like a skin cell, right, versus a neuron.
Yeah.
They are in different, they have the same genome, right? But they're executing different functions, and they're clearly in different states, right?
Absolutely.
And so then you could go and look at their collection of RNAs. We call that the transcriptome, because each RNA is like it's a transcript, and the collection of all RNAs is the transcriptome.
Right.
And so you can look at the transcriptome, and then you can see basically the steady state difference in the transcriptome between neurons and skin cells. And you'll find that while there are some genes that are the same, right? Like there's a bunch of genes that the cell just needs to be around to like live.
Yeah.
Right.
Right.
But then you'll find a bunch of genes that are like neuron specific or skin cell specific. You'll find like neurotransmitters. You'll find like genes that encode the function of the neuron.
Right.
Right.
Yeah.
And we can actually think about those, like you can look at the correlation like structure of genes that make up a neuron versus a skin cell, and you can actually assign like modules of genes, right, like that form circuits that make a neuron. And then one really interesting question that challenge in engineering is like, oh well, maybe I want to make a skin cell into a neuron, right? Because they have the same genome. So I could imagine like, and you can do maybe converting the skin cell into the neuron, and then you might say like, oh, what parts of its transcriptome do I need to perturb, like move, right? Like if I could just move the state machine from this state to that other state, does the skin cell turn into a neuron? Well, I guess it's like a little bit more complicated than that, because the state is like memorized by something happening in the genome, and we can talk about that separately. But the genome has some memory, right? It's not like yeah. So there's this whole field about thinking about like how do you reprogram the memory in the genome so that you can convert like one cell type into another, so they end up in the same state.
Is this the idea basically kind of with like Yamanaka factors?
Yeah, like Yamanaka factors. So what do Yamanaka factors do? It's actually interesting. So like you can take a skin cell and you can add these transcription factors, and it converts the state of the cell into something that looks like a stem cell.
Right.
It's some undifferentiated cell, and what it's doing is it's kind of like erasing the memory the cell has of where it's supposed to be. Right.
Yeah.
I guess like an analogy would be like, it's like ROM, read-only memory.
Yeah.
The cells are like, you like program the cell. The cells are like stuck in the state, but you can like flash ROMs. You can erase the memory. Cells are not like RAM. You can't just like move the cell from state 1 to state 2.
Yeah.
They're a little bit like ROM. Like you can have a skin cell and it has like the ROM for the skin cell. And the Yamanaka factor is like flashing the ROM to convert it into this unwritten state. And then you can like flash it with the neuron. And that's what people are doing. Although there's a very interesting idea of like, can you transdifferentiation, which is like, can you take skin cells which are like stuck in this, in our analogy, they're like this read-only memory for skin cells. Can you just like change that memory somehow directly into the, the state
without going back?
Without going backwards.
Interesting.
But going backwards is the way that we have learned how to do it the most traditionally.
Is there a problem with going back? Like, it seems more difficult to go straight.
It is much more difficult to go straight, and it's unclear if there's ways to traverse that landscape. You can imagine. It's unclear if you can move between those two things, but there's a lot of, I think, benefits to going straight in the sense that, like, you might be able to do it in situ. Like, if you, like, had some injury. I mean, people are interested in that, right? Like, if you had some injury to your spinal cord, like, could you go, like, convert the nearby cells? Or, like, there's a lot of places where you can't deliver cells, right?
Yeah.
So you might want to do, like, in situ, like, transdifferentiation from one cell type to another.
Okay.
Just because it's hard to, like, take cells out of your body and then convert them and then put them back into the places that you might want to put them.
Right. Yeah, absolutely.
So
this is something that, I don't know, maybe will illustrate my lack of understanding currently, but you have, two cells, they have the same genome, but one's a skin cell and one is a neuron. And there's this idea that an analogy of, there's kind of the ROM basically. Where in the cell is the ROM? And
right.
Then, you have the same genome, so
yeah, exactly.
Where's the ROM where is like the ROM existing, basically?
It turns out, and this is just some of it, like a really interesting question. Like the ROM is stored in, it's like this persistent state that's stored in like the proteins and marks. So there are these modifications to the genome, like chemical modifications to the DNA that help store the state.
Okay.
The classical one is this thing called DNA methylation.
Okay.
And that's like where a methyl group gets added to one of the cytosines, and it's just like a way to like mark the genome. And those marks get persisted when cells divide, and by some very complicated maintenance machinery. So that's one aspect. But then there's also like, it turns out that you have this 3 meter long. So like if you stretch out the 3 billion bases of the genome, I think it's like, maybe it's not 3 meter, maybe a meter long linear sequence. And you pack it into this 10 micrometer sphere. But it's not randomly packed. It turns out that basically the 3D packing of that genome determines what genes are maybe turned on and what genes are turned off. So there's like a structural element. There's like proteins which maintain like the state, and then there's probably positive feedback loops. That maintain the state. And so it's like some combination of all of those things, and that's why it is complicated to figure out how to do this. This transdifferentiation or any sort of these things is that we don't really understand. That's like the question you asked is not naive at all. It's like actually like a pretty fundamental question. It's like, you know, the difference between these two cell types, like where is that information stored?
Right. Got it.
And we have a broad strokes understanding of like what types of substrates it might be stored. But if you were to ask me, where is the information stored between a skin cell and a neuron, I would be hard pressed to give you like, you know, these are the parts in the skin cell, like genome, which are methylated, and these are the proteins that need to be around. It's this like, and so, I mean, it's a kind of a fundamental question.
Okay. Yeah. So with this idea of kind of the 3D structure being, you know, it's not trivial. It's important to what actually becomes a cell. Is that part of the reason why stuff like expansion microscopy and like understanding the 3D structure of
for sure. I mean, part of my research, our research, is trying to understand the 3D structure of the genome and how that relates to basically genome function and what genes are on and what genes are off. And you know, I think it's, I mean, here's like the fundamental thing that's also crazy about it, right? Like we just posited that the structure of the genome was important. Well, like every time the cell divides, you like randomly you have to like condense all your chromosomes. You have to copy your chromosomes, condense them, and then reassort them into 2 new cells, right? And if I just said that the structure of the genome is important for maintaining what cell type I am, well, that means that the skin cell, right? Like every time a skin cell divides, it has to maintain those physical structures, right? With high fidelity, in this, like, you know, packing 3 billion bases into this really small volume. Right? So, I mean, that's just kind of a very amazing act.
Right. Yeah, absolutely. So, does your group, or is there interest kind of more broadly even in like measuring this across time, or is it always kind of a static? Static
definitely. We're very interested in measuring things across time, and I want to point out, I mean, Anders Hansen, who's like right there, I think, actually. He's a MIT BE professor that we collaborate with. I mean, one thing that we're very interested in, his group is very interested in, is like taking videos of like individual, like just label 2 points in the genome. And just taking like dynamic videos of how they're interacting and moving around. And we are very interested in temporal measurements, but it is challenging because almost all of the measurements that we have in biology involve like killing the thing that we want to measure. At least molecular measurements. I mean, obviously there's like non-invasive measurements But molecular, if you want to ascertain the molecules, like what protein or what gene or at what mRNA, they're almost all destructive.
Yeah.
Although, we're very interested in ideas for like this idea of molecular recording. I mean, I just told you that like we're really good at reading the sequence of DNA or RNA.
Yeah.
So if we can convert, we're interested in ways of converting dynamics into something like sequence. So like, here's like one idea. This is not this is an idea I worked on in undergrad and something that George was really, not undergrad, in grad school, and something George was really excited about back in the day too. George Church.
Yep.
It's like, one area of dynamics you might care about is like the activity of neurons.
Sure.
Right? The firing of neurons.
Yeah.
And imagine if you convert when a neuron spikes into like one base in a piece of DNA, like an A.
Oh, okay.
And then when a neuron is like not spiking into like another base, then that's a temporal record in sequence. And now you can like then sequence that and reconstruct like the history of that neuron's firing. And the thing is that sequencing is very scalable.
Right.
But there's other ways of doing it. You don't have to encode that dynamic information in DNA. You could encode it into other substrates. I think Adam Cohen and Ed both had a paper that was very interesting in bioRxiv like a couple months ago, where it was like encoding it in like a protein tube. Like it was basically like a ruler. It's a thing that continuously polymerizes at a set rate.
Okay.
And then like if things were happening, if the cell was in one state, it would be read. It basically makes like a binary barcode in your cell that's like a physical structure.
Yeah.
And then you can read it out later by a microscopy.
Okay.
Right?
Yeah.
And so it's like trying to figure out ways of converting dynamics into permanent records.
I see.
Yeah.
So is the idea that you mentioned with George, is that the thing that's called ticker tape?
Yeah, that's a ticker tape.
Yeah.
That's like a molecular ticker tape. And then there's been a lot of other ideas so that's a much harder idea.
Yeah.
There's been the best implementations of molecular recording have been for lineage tracing, which is like understanding which cells are born from which other cells.
Okay.
Because you have like this is another amazing part of like biology is that like every cell in your body came from like one cell, right? So for organisms like C. elegans, you can like just like look on a microscope and see like which cells became which cells. And like somebody did that in like the 80s. Right?
Okay.
Like, they made an entire map of like because there's only like a couple thousand cells or something.
Yeah.
But you can't do that for like mammals or and so there's been some cool ideas where you go and you make like random scars. If you just introduce like random mutations in the genome at a set rate, you can actually like reconstruct the lineage by looking at like basically like it's like 23andMe for cells, right?
Yeah.
Because like you can connect the relatedness of two individuals by their like shared mutation.
So it's kind of like the thing with calico cats, but
yeah.
Many dimensions, not just your
exactly the kind of thing with calico cats. So if you just introduce a random mutational process, then you can look at like the shared mutations, right? Because like the daughter cells will all have that shared mutation plus additional mutations. And so that's the fundamental concept, and there's been like implementations of that concept with different enzymes. CRISPR-Cas9 is being like one prominent one.
Right.
So that's one version of molecular recording, and that's at a really long time scales, right? Like in the sense that like there you need to make one record every time the cell divides. At minimum, maybe. But there's like probably like we worked on a project where we try to determine the age have use the same idea where we accumulate mutations on RNA, and now we can determine the age of a given RNA just by how many mutations it has. It's like a clock, right?
Yeah, absolutely.
And I mean, that could be cool in the sense that, it is cool in the sense that you can use that to reconstruct when the RNA was made. And a lot of genes are made in response to stimuli. And so you can then try to reconstruct the past dynamics of stimulus, right, through that. So I think there's some ideas about doing molecular recording, this broad field of getting dynamics. The other thing would be way to get at dynamics. Another aspect of that molecular recording before I finish is like interactions between cells, right? You might want to know when two cells have like seen each other. If you have like dynamic migratory cells or cells that are moving around. We're interested in this problem, but I think other people, people have invented like tools wherein like you can transfer a label from one cell type to another cell when it sees it. So that's another way of looking at like the history, right? Like here you're like marking interactions, and at the end you see which cells have interacted. Right? So those are like ideas of like converting something that happened in the past into a permanent record. And then there is probably another set of things for dynamics where you can try to infer causality from perturbations. So like that's actually how we learned the most of what we know about a lot of biology is that, you instead of looking at real dynamics, you're looking at like, I'm going to like get rid of this thing and then I'll know if this thing is responsible, causal, or like necessary and sufficient for this other thing. And that's how we map functional genomics, where we like map genes, like what their functions are by like just knocking them out. I think perturbations are going to be quite exciting for inferring causality. It's another area where you can get at dynamics.
So if I can ask you about some specific things from your lab.
For sure, yeah.
I guess that we've kind of covered some of the spatial genomics and temporal genomics stuff. One thing I saw on kind of your homepage that I wasn't able to really understand in the time that I had, which is this thing called Slide Seq. Slide sequencing, I guess. Could you tell me about that?
So we talked a little bit earlier in our conversation about how, when I started my lab at the Broad, I wanted to mirror I wanted to bring together this world of like sequencing the compositions of cells, right? What messages they are expressing. And the world of microscopy, where you can like look at where the cells are or who their neighbors are, but you can only look at like 6 of those molecules at a time. And so Slide Seq, the concept is really simple. It is, imagine if I like made a camera, right? Like a camera is like all composed of an array of pixels. And instead of taking pictures in photons, the camera is just like capturing these messages. Like each pixel is capturing what messages it saw. These mRNA molecules. And so how we make this camera then is that we take beads, like each pixel is a bead, like a microbead. It's about 10 microns. And
what is it? It's
made out of polystyrene.
Oh wow! Okay.
It's a polystyrene microbead.
A physical little microbead.
And on it there's a bunch of barcoded DNA. And what I mean by barcoded is what I mean that like each bead has a unique clonal sequence. Each bead has a unique sequence of A's, C's, and G's and T's on it.
Oh, I see.
And what we do is we determine the XY. We determine the relationship between we put these beads out onto like a surface, and so it basically forms like a an array of pixels.
Right.
And on each pixel, we now determine like the XY location and also like the unique sequence that's associated with that XY location.
Quick question. So you have a bead. Is there one barcoded DNA per bead, or is there many?
There's about 10 million
Okay.
DNAs per bead.
And do they all have the same barcode?
They all have the
bead?
same
Okay.
barcode.
Okay. So it's basically just an identifier that says what bead it is.
Yes, an identifier what bead it is, and then we associate the identifier with which spatial location it's in.
Yep.
So now we have this like effectively this pixel array, right?
Yeah.
Because we know this pixel array that's like where there's a correspondence between position and barcode.
Yep.
And then what we do is we take like a piece of tissue, like let's just say, there's you can take like tissue from pathology or animals or whatever.
Yeah.
And what you do is you can actually freeze it.
Okay.
You freeze the tissue into like a block of basically ice, and then in this like fancy like deli slicer setup thing.
Okay.
It's a glorified deli slicer. You can cut a very thin section.
How big are we talking of? A little is this a microscopic piece of cell, or is it
no
like a
so
millimeter?
we will take like a mouse brain.
Okay.
Right? A mouse brain is about
wow.
a centimeter cubed.
Okay.
Okay?
Yeah.
And you freeze it.
Wow.
And then you
Okay.
mount it onto like this deli slicer. Wow. And then it sections like it just makes like a section of it.
Okay.
And you can make a 10 micron section.
Oh, okay.
Which is about like a layer of cells.
Okay.
But it's still a centimeter by a centimeter.
Okay.
Right?
Wow.
So it's like literally a 2D plane.
Yeah, absolutely.
Of like the organ.
Right.
And you lay that 2D plane on your pixel array.
Okay.
And now we take the transcripts, the messages. The messages from that 2D plane is captured onto the beads. And
so, sorry to interrupt you again, but the bead, how big is the bead?
10 microns.
10 microns, and you have a 1 centimeter cube, so you have like 10,000 squared pixels. Is that right?
Yeah, it would be 1 centimeter. Yeah, if 10,000 squared pixels.
Okay.
Usually we do 3 millimeter. We usually have 3,000 squared, about 100,000 pixels.
Okay. Oh, okay. That's pretty manageable. It sounds like.
We have like a 100 kilopixel, a 0.1 megapixel. No, that's a 0.1 megapixel.
Yeah, 0.1.
Camera.
Okay.
Okay. But the messages from the tissue get transferred onto these pixels, and they get barcoded. They get associated with the unique sequence of A's, C's, and G's and T's.
Got it.
At each pixel.
Okay.
And then now we put that onto like one of these high throughput sequencers, and now we can sequence the barcode and what the message was.
Oh, I see.
So now you're just like, it's like effectively just, as I said, it's like the analogy of the camera is like very apt. Now instead of taking pictures of photons, you're taking pictures of these like messages.
Right. Yeah.
And that's cool because now like it's like a microscope with 10 micron resolution.
Yeah.
But now you reconstruct all of the messages
right.
from each cell, and as I just said, like the messages tell you something about the state of the cell. And so now you can see and at the first level, 0th level, you can like look at where the genes are being expressed. And then at the next level, you can group the genes based on like what you know about cell types, right? You'll see a bunch of neuronal markers that are co-expressed that are all captured by this bead. You have like a good likelihood that there was a neuron here. And actually we have much higher resolution than that because you get to look at the whole transcriptome. So we can basically classify where cells are on these beads.
Okay. And basic kind of technical question that just for clarification. So you have, for example, in the mouse brain, you have, is it RNA or mRNA? It's not important.
It's not that important.
Okay.
We're capturing mRNA.
So you have mRNA and you have little beads with marker DNA on it. What exactly happens? Does the RNA go onto the beads, or does the DNA go onto the...
The RNA goes onto the beads.
Okay.
In a diffusional type. It's just like a diffusion thing.
Okay.
And then there was this process where we associate the DNA with the RNA in the sense that we use this enzyme called reverse transcriptase.
Okay.
And what it does is it takes it uses this barcoded piece of DNA as a primer, and it synthesizes the complementary DNA of that RNA. And so now you have this one molecule that has like the RNA sequence on it, and also and
that's how you get the
exactly.
Okay, wow. Okay, that sounds pretty awesome. And then you take this. Do you? Is there kind of just a thing where you kind of like wash out the RNA from the styrofoam beads? Or?
So after that you basically you don't have to keep the pixels intact anymore. You can just collect them,
because you have the XY data.
Yeah, and I said that you basically use this PCR, which is this way to amplify. You amplify all of the molecules that you captured, basically.
Yeah.
And then you put that onto this other machine.
Okay. That's pretty awesome. Yeah. So kind of from a higher up perspective, what what are your like kind of more broad goals over the next, I don't know, 10 years, 20 years? Like what would be a successful next 10 years for you? Yeah,
I mean, what we've done so far in the lab is like come up with ways to measure the organization, map the organization of biomolecules, whether that's like where genes are being expressed in tissues or where cells are.
Yeah.
Or at another resolution is like, what is the organization of the genome?
Mm-hmm.
Like, I'm really big into so one of the things about biology that's hard compared to engineering is that there are not good abstraction layers.
What do you mean by that?
In the sense that if I'm an engineer and I'm building a circuit, or let's say I'm if I'm building a circuit, I don't have to worry about how transistors work.
Yeah. Okay.
Right. I can take an op amp and whatever, and I can build my circuit with an abstraction. I don't have to worry about E&M when I'm building circuits. But the abstraction layers are really fuzzy in biology.
Oh my god.
If I want to build a tissue, I can't use cells as abstractions because cells have weird interactions with each other, or we just don't know the rules.
Yeah.
Right. And so one thing that I think would be really exciting is coming up with the rules, learning the rules of tissue organization. What are the interactions that are important, and what are the molecules that are important to form function in any tissue, like the brain or the kidney, and being able to then manipulate these rules in an abstracted context, take those rules and recapitulate them in models where you put different cells together, tissue engineering, where you try to build tissues, right? I think success would be if we can take the rule, if we can understand the rules of tissue organization such that we can actually go and build new tissues with new functions, right?
Yeah.
And so that's one aspect, and the other aspect is similarly for genome organization, right? If we could understand gene regulation in a way, where we understood the rules of how structure affects function, and that we can reorganize the structure,
right,
to change how gene expression, or if we understood that fundamental question of what is the memory states, right? And then we might be able to then reprogram the memory states.
Yeah.
And so a lot of what I think about is how do we generate abstractions for biology or maybe we can't generate them, but we could at least come up with frameworks for thinking about how to engineer biology, right? And we're doing that through building computational models. We're doing that by developing new ways of making measurements, and then we're doing that by making perturbations in those models. We'll make predictions about what perturbations to make, and then we'll make those perturbations, and then that will tell us how correct our models are. Right?
Yeah. So I had a question for you about kind of the state of the field as a whole, or maybe not even, maybe it's larger than the field, but there's people who are, working on various anti-aging stuff, various gene therapies.
Yeah.
And it's not, as an outsider, it's not clear to me that we know enough to be successful yet. And it seems like your work is really useful to get us to actually understand what's going on a little bit better. So I guess the question I'm asking is, do you think that more work should be focused towards kind of developing tools for understanding what's going on like you're doing, or do we need both? Like
I think we need both. Actually, I think we need both tools to understand what's going on, and then a lot of characterization of what's going on to like, I think there are efforts to do that. Mm-hmm. I heard about some cool efforts to do that where it's not focused on perturbations to like, it's to investigate published perturbations and figure out what exactly is going on, right? Yeah. Because, there's a lot of excitement about, for example, like delivery of Yamanaka factors, but I think there's a lot of characterization that needs to be done about like what are we actually perturbing in these systems, and I think there's so much to be learned also in just understanding like what is normal aging.
Yeah.
Right. And what is pathology.
Yeah.
And what is like a healthy tissue.
Mm-hmm.
And versus what is like an aged tissue, whatever that means, right?
Yeah.
And that's, I'm not like a huge aging person, but like, that's how, like one thing that I've been thinking about is that like, we need to come up with like a language to describe what are the phenomenon, right? And there's lots of ways to describe what the phenomenon are, but it'd be great if that language could be used to make predictions that we can then and go and test, right? So that's one thing I think the field needs a lot more of. Just like that, I guess that's what I'm saying. Like that's why we're doing these things. Is like how do you characterize the rules and the frameworks that these things are operating under?
Yeah.
But I do think it's exciting that so many people are into it. And you get the sense that like, a lot of biological discovery is serendipitous, right?
Yeah.
And so,
of course.
You get the sense that like there might, I think you get the sense that you have to be in the right place, right?
Yeah.
And then you're serendipitously find this thing, and we might be at the right place. There's some excitement that we're at the right place, and so more efforts to try to increase the like interaction frequency of serendipitous events will be good.
Yeah. Huh. Okay.
Like, I guess what I mean is that, oftentimes things are obvious from hindsight, but you haven't been in the right framework of mind to think about it. Like expansion microscopy, like, it would have been difficult to develop that a long time ago, even though it was wholly possible, because people weren't thinking about problems at that length scale, and you needed the development of certain like molecular tools to make it interesting, to like really good antibodies or really good fluorophores, and then I think it requires kind of being at the right state in the field for you to think about the impact of the problem, right? And so that also happens in other, I'm sure that happens everywhere, and so there might be some sense that we might be at the right place.
So I'm not sure if it'll be useful, but like I don't know, I find like my entire time in college, like the most useful conversations I've had, like the most useful thing I've had from college isn't stuff that I could have learned from textbook. It's talking to people. So along the ideas of kind of serendipity and kind of like being at the right place at the right time with the right ideas, is there anything that you do kind of outside of work that kind of puts you in the right mindset or kind of helps you
Yeah, that's a great question. I love reading science fiction,
Oh, really?
actually.
Oh, wow. Okay.
Or I used to, I love reading science fiction, and
Yeah.
I used to have a lot more time for it. And now I have not recently, but
Sure.
I was a big fan of the classic, science fiction, Asimov,
Sure.
Dune, these sorts of things.
Yep.
But I would say for a while, I really enjoyed reading Neil Stephenson. I think he's got some very prophetic things that happen in his books. Like Cryptonomicon, prophesized the cryptocurrency revolution, maybe. But why do I think it's interesting? I think it's just interesting to a lot of those books like also explore like society in interesting ways. I think beyond just it's like a way of doing thought experiments about society,
right?
Which I think is quite cool. I think being at the right place at the right time. I think one thing I do like doing is I love talking to other people in other fields about their science.
Yeah.
Like astronomy. Like
Sure.
I'm super into talking to, I don't know that much about astronomy, but I like love talking to astronomers about what they're doing.
Yeah.
In, like these deep sky surveys.
Yeah,
like
absolutely.
Exoplanet,
Right.
And I can't tell you of a time when there was an idea that I talked to someone else about that became really relevant in my research, but
Yeah.
I think it's helpful to just there's a lot of that awe and wonder about learning about new things that are way outside of your field that that helps just you be more creative, I think.
Yeah, for sure. So you kind of mentioned, talking to people far outside of your field. One thing I wanted to ask about kind of talking to people in the field was I actually happened to find online that a while ago you signed up for something called OpenWetWare, which was from what I was able to see, it seems like kind of a defunct webpage now, but it seemed like some organization like the early 2010s maybe to try and kind of pull together people.
It was, I think maybe here at MIT.
Oh, yeah?
It was for synthetic biology.
Yeah.
And, here's what it is that, it's actually for this exact same reason of, there's not really good abstractions for engineering biology.
Exactly.
And so, it was a way for people to characterize and build parts that could maybe be put together. And it was part of this program called iGEM, which is still around. I mean, it's really, really popular these days. It's like this international genetically engineered machines thing. I mean, I did that as an undergrad, and that's why there was this OpenWetWare page.
Yep.
I mean, I think that is and continues to be a great thing because it inspires a lot of folks to come into biology from other areas.
Yeah.
I mean, speaking about that, I think it's interesting to think about how organizations are set up.
Yeah.
Right?
Sure.
I'm sure you guys might have thought about how do you set up organizations to change to really generate impact in science.
Yeah.
Right. Whether that's motivation, how do you generate the right incentive structures in terms of grants and how funding is distributed. I think that's a really interesting aspect. Mm-hmm. There's also just how do you build organizations to disseminate research, right? And perhaps interest populations of people that you couldn't access before, right?
Yeah,
and,
absolutely.
I think it's very interesting to think about those questions.
Yeah.
And I think the cool thing is I think there's a lot of innovation happening in that space right now of how do you think about new models for science. I'm helping out with this new science program, and it's a way for it's like a fellowship, right? Basically for people who have some ideas about what they want to try in biology, but they're not like in a PhD program necessarily.
Interesting.
I think that's but they want to pursue some independent idea for a short amount of time just to get the feasibility.
Yeah.
I think there's also like a lot of innovation happening in funding. The Broad Institute, right?
Yeah.
Was like super innovative, I think, back in the day when it was formed, because it brought together this idea of like really large science, right? I think there was this thing that happened, I think, like Vannevar Bush, like when he set up the NSF in the 50s. Yeah. That he transitioned, there was a strong emphasis on investigator-led science where like, which is important. Like, how do you like generate, like everybody has their own ideas about what is really they're passionate about.
Yeah.
And they're all like a bunch of basic science.
Right.
There's a lot of basic science questions. And that's a lot of how the scientific system, funding organizations are organized in the United States.
Yeah.
And that's great. I mean, we really need that investment in basic science research.
Right.
And that's really apparent in biology. But if you look at physics and astronomy, there's also this extreme aspect of team science, right?
Yeah,
where
absolutely.
you like tackle these really large problems.
Yeah.
And that like didn't really exist in biology. I mean, there was the Human Genome Project, but I think like that sort of philosophy is a little bit antithetical to, not exactly antithetical, but it's a little bit different than this sort of investigator-led, like, basic science directions.
Gotcha.
And I think we need both of them. And, when the Broad was set up, there was some idea about doing these like large-scale science projects with bringing in lots of expertise and, the community, like focused on human health and disease. And that's been really successful. And I think there's a lot of experimentation now about like, how do you empower such efforts? Now that model is successful, right? And you see quite a lot of different like types of organizations being built.
Yeah, for sure.
Whether they're for-profit or non-profit. I think, there's a lot of money in investment in biotech right now, right? And there is some interesting questions about basically like how you set up the model in a way that it's it's both accessible and successful for the scientific community and people, but it can also tap into like, both the translational aspect of venture, but also like the fact that a lot of the successes in biotech are building off of like basic science research or like these sorts of innovations that happen at research institutes that don't quite get recaptured into the basic science endeavor. Right?
Yeah, for sure.
So I'm sure there are like lots of really innovative ways to align those worlds such that like everybody wins, or there's just more things that happen. Yeah,
we're for sure seeing a lot of that, which is nice to see. Last question, just because I know we got to let you go, which is if you could give advice to your younger self, or maybe give advice, broadly writ to undergrads or grad students, do you have any kind of words of wisdom, or even high schoolers kind of?
Yeah, that's a interesting question. I actually think that one thing would be to I think if possible, go out and talk to like people. Like if you're excited about something or like many things, go out and like reach out and talk to like the people doing research in that area. I mean, I think it's easy when you're an undergrad to be very focused on or high school. You're like everything is, you're like super focused on the now, right? Like on coursework or on like, even if you're doing research, you're focused on that project.
Right.
But I think, undergrad is like just such a special time for you to just take up like so many new ideas. And also you have kind of some access, right? You're like oftentimes at institutions where there's like really great people doing really great science. Yeah. And they'd love to talk to you about it if you just even ask them. And asking them what the big questions in their field are, and just getting a sense of like the bigger picture of the field or arc of science that you're interested in.
Yeah.
I mean, I feel like you never take that. I mean, I certainly didn't take that seriously in undergrad, because you're always just so focused on.
I certainly was.
And on the things at hand, and being able to have that like introspection to like go back and take a step and just see the patterns. And if you do that, maybe like you can identify what's really important, or what are like the really interesting questions, and that will really set you up.
Yeah.
If you want to go, and well, no matter what you do, if you do science or if you do technology or venture, like I think it's all going to be really important to get that bigger picture view.
Yeah.
I think it is true. Like, it just, when you're an undergrad, you just never.
Oh my God.
You're like you're like always focused on problem set, or
I deeply regret it. I just maxed out on classes every single semester.
Exactly. And you're like super focused.
Yeah.
And you do research, but you're like, oh, I really want to get this research project to work, or like I need to get this next experiment to work.
Yeah. Even in grad school, up until the past year or so, it's just been.
Yeah, I think it's just super, and like, I think also like the system selects for that,
Yeah.
Right, like that's how you become successful, or like by whatever metric you're successful, like the system like selects for people who are good at doing that, right?
I'm drastically behind in physics because I've stuck my head out. I'm like, oh, like maybe bio's kind of maybe an interesting field, but it's like you'd be screwed if you want to go back into physics now, because you're behind by a lot of time.
Yeah. So, but I do think it's important to, because you just don't. Also, like, oftentimes your path is so like your path is so like path dependent, like.
Yeah.
And
absolutely.
You might want to like get rid of that path dependence a little bit by.
Yeah, that makes sense. Yeah. Awesome. All right. Well, I'll say thank you so much.
Yeah. Thanks.